Hello, everyone. Thanks for having me. The title is Temporal Pole Amygdala Dynamics in Affective Picture Viewing. This is a Stereo-EEG study. Okay, so mapping emotions to the brain. So the first thing that we think about is amygdala. Then there are other structures, limbic structures, orbitofrontal cortex involved. But one region which is widely connected to all of these regions and is involved in some of this functioning, but often receives less attention, is the temporal pole. Now, the evidence for the socio-emotional functioning of these regions comes from mice studies, lesion studies, fMRI, MEG, and EEG. But often there are constraints with some of these techniques, such as EEG source reconstruction is often difficult with substantial variability, and fMRI both signals is compromised from deeper structures. And especially in case of temporal pole, the signal is distorted because of magnetic field inhomogeneities. So how do these regions respond and interact in affective picture viewing? To address this, we used data from intracranial EEG with our collaboration from an epilepsy clinic in Brisbane. So Stereo-EEG is the implantation of depth electrodes in different parts of the brain to find the epileptic focus in patients with intractable epilepsy. So you can see the picture at the bottom, the X-ray of these electrodes shown in one of the patients and rendered onto an MNI space of the brain for all the channels and electrodes. So we had 6 patients in this study, and we tested them on a cognitive paradigm. To localize the electrodes, we used a preoperative MRI scan and a postoperative CT scan. You can see all the channels in the CT scan in the bottom image. And then these were co-registered into MNI space and getting us the electrodes that we were interested in. And this was assisted by the implantation maps provided by the epileptologist for the clinical evaluation. We ended up having 1 channel each in temporal pole and amygdala. And we had a total of 4 temporal pole channels and 4 amygdala channels. We had to exclude 2 participants, 2 patients, because of some abnormal data found in MRI and the data recordings. Okay, so what was the task? We used the classic affective picture viewing paradigm, which had different pictures of high valence, neutral valence, low valence taken from the International Affective Picture System. Okay, moving on to analysis. We first performed some time frequency decompositions and found really broad range frequency activity in both of these channels. Now, this high frequency activity is an informationally rich and temporally and spatially precise marker of neuronal activity. And we quantified this high frequency activity with high frequency broadband amplitude, which expressed as a change from baseline. And we see this time-locked responses in both of these regions, showing that there's increase in activity while viewing these pictures. Moreover, the mean of these responses was highly correlated to each other, showing that these were responding consistently and to these pictures and correlated to each other. Then we moved on to some functional connectivity analysis of coherence. Now, coherence is a measure of synchronicity between 2 signals and tells us about what frequency range is the communication in. So after performing this coherence, we see a peak in low frequency range where the communication is happening. And this was not much different from the baseline condition, perhaps telling us that this is what the intrinsic frequency of communication is between these regions. Now, to know the direction of communication between these 2 regions, we did something called a spectral Granger causality, which is basically extension of Granger causality in time domain to frequency domain. So it tells us the information flow in what frequency range is happening. And we found that this information flow is predominantly from temporal pole to amygdala, and not from amygdala to temporal pole. And the frequency is, again, the low frequency from 0 to 10 hertz. This was statistically tested with permutation testing. Okay, so far I've talked about all the direction and communication with all the conditions combined. Now we wanted to know how the effect of the modulation condition is on this connectivity, for which we use dynamic causal modeling for phase coupling. Now this DCM for phase coupling is a phenomenological model which models the data feature, which in this case is the phase of the signals. And we restricted our frequency range from 2 to 12 hertz based on our previous coherence and spectral Granger causality results. And we built our model space, including all the possible connections and affect modulation direction, and ended up having 8 models to be tested. After performing DCM, we found that the model which was consistently winning was the first model, in which the affect modulation was from temporal pole to amygdala. And interestingly, the second and the third model, best models, which were winning, they also had the same feature, suggesting that there's strong influence of temporal pole on amygdala. Now, with these results, I'd like to draw upon 2 conclusions. Now, temporal pole's influence in affective information processing to amygdala can be explained in the framework where temporal pole acts as the semantic hub or the conceptual store of knowledge and is involved in emotion recognition, emotion identification, integrating contextual information, and perhaps even generating emotional response, which is further appraised in the amygdala. And such a role is also consistent in emotional disturbances, which are found in variants of dementia, semantic dementia as well. Secondly, the high frequency activity found in these regions and the communication in the low frequency range gives credence to a communication through coherence hypothesis. So for example, here, the 2 neuronal groups, the red and the green, the high gamma burst activity is happening at excitability peaks of each other, and that's why there's coherent communication between them. Whereas another neural group, shown in black here, although has the gamma burst activity, but fails to communicate efficiently because it misses these excitability peaks. Okay, with this, I'd like to wrap up. Thank you for your attention. I'd like to acknowledge my supervisors, Christine and Michael, and other members who helped me with this, especially Sasha Dionisio, who's the epileptologist with Mater Hospital. I also have a poster, so if I have missed some specifics and you want to chat more about this, please come to my poster. Thank you, and I'm open to questions.